MECP2 duplication syndrome was discovered in 2005. It is a progressive neurological disorder primarily affecting males. Common features include nonverbal communication, infantile hypotonia, low muscle tone, recurrent deadly respiratory infections, and epilepsy. Other signs and symptoms include ataxia, wobbly movements, reflux, aspiration, and feeding difficulties. Recent technological breakthroughs in communication have shown that children affected by MECP2 duplication syndrome have been essentially trapped within their bodies. In the very recent past, a combination of non-existent or limited speech with poor motor skills have caused these adolescent individuals to present in testing as having severely delayed cognitive abilities. Parents of these children knew that traditional assessments were not capturing the brain power of their children. Pioneering communication, work with MECP2 duplication children, and eye gaze communication devices have begun to validate the parents as it was demonstrated that these children have the ability to communicate requests and feelings. In fact, Bo Spangler has been a leader in this field and has been featured in international press releases. The genetic abnormality that causes MECP2 duplication syndrome is a double dose or duplication of the MECP2 gene. The protein made by the MECP2 gene, called MECP2, plays a pivotal role in regulating brain function. Too little or too much of the MECP2 protein results in brain dysfunction and physical impairment. Mutations in the MECP2 gene are also commonly associated with Rett syndrome in females. Advances in genetic testing has led to increased diagnosis of MECP2 duplication syndrome. Studies suggest that prevalence may be 1.8 per 10,000 live male births. 50% of cases do not live beyond 25 years of age. But there is great hope for a future without MECP2 duplication syndrome. In 2015, Dr. Huda Zoghbi published an article showing that not only is MECP2 not neurodegenerative, but a complete reversal of symptoms using gene therapy is possible.
We're very excited, first of all, by the work that we were able to do thanks to the support of the family and the 401 project. What we have been able to do is answer the question, is the duplication syndrome reversible? How do you know if it's reversible? What you do is you let the animals develop all the symptoms that we typically see that are very reminiscent of the features we see in the human patients. And after they developed all the symptoms, we take one of the extra genes away genetically. We turn it off, and once we turn it off, we get back the mouse to have just one copy of MECP2. And what we saw with time, as we did that, the animals got better and better. And basically all the symptoms we saw in them with the duplication slowly disappeared. So this was very encouraging. This was very exciting to us. The brain can recover when you take away the extra copy of the gene. What this did, it then inspired us to explore a new potential therapeutic approach that we had not considered before, which has gradually been tested in some various models of neurological diseases to regulate levels of proteins or to really target specific genes within a cell. And that technology is called antisense oligonucleotides, which means they're little pieces of DNA that can bind to a particular gene and lower its level. And in this particular case, we wanted to test if we can use this little piece of DNA that we can deliver into the brain and see if we can lower the levels of MECP2. Why did we want to test this? We wanted to test this because this is something you can test in human patients. Genetic rescue, where you manipulate the genome, is not something we can do in humans. So we really needed to move from things we can only engineer and do in mice to something we can now perhaps one day do in a clinic in a patient. That's why we tested these antisense oligonucleotides. And once we established they can do so, we then implanted them in a little cannula. We gave them via the ventricle because you have to get it to the brain. This is not something you can give in the blood and it'll get to the brain. It took a few weeks for the protein level to get back to normal. But the very nice thing is that as the protein level stayed normal for about 4 weeks, we gradually started seeing the symptoms go away and the animals improved. What we will have to do now is make this mouse now with both of its copies coming from the human gene to titrate, and we'll have to test a lot more of these small DNA pieces to find the perfect one with the minimal toxicity. Once we have that established, we will then go to a non-human primate model to test it further, and only after that we will be ready to move things into the human. So this is one of the projects that was funded by the 401 project that yielded very exciting fruits that we have now a path forward to follow.
So where are we now? Multiple approaches are being studied by researchers globally to develop a drug to treat MECP2 duplication syndrome. For this exciting ASO therapy, Dr. Zobi and her group continue to complete preclinical experiments critical to bridge the gap between the lab and clinical trials. This research is currently being funded by RSRT. Before this event, families have $150,000 out of the required $300,000 to finish these studies. With the help of your generosity at this Cure MECP2 event, we hope to get much closer to meeting the required $300,000. We are confident that Bo Spengler and all the other people worldwide affected by MECP2 duplication syndrome will have a future free from this syndrome. Bo Spengler doesn't give up, so we don't give up either.